Hi, my name is Veronica Cappas. I am a biology and forensic science major. I play soccer here at Piedmont College also, and I am attending Penn State to pursue my master's degree in forensic science next fall. My presentation will be over the potential development of a postmortem interval estimation using microbiome changes during human decomposition. Before I begin my presentation, due to the nature of this research, there are graphic photos within this presentation. First, I am going to explain what the postmortem interval is, or PMI. It is basically the time of death. It is important in all matters of death investigations. It can create a timeline of when the crime was committed and eventually help catch the murderer. It assists with the cause of death. There are 5 types of death: natural, accidental, homicide, suicide, and undetermined. It can also assist in civil cases, deciding who inherits property or if there's a death policy in place at the time of death. There are many different methods used to determine postmortem interval. The first is pallor mortis, or simply the whitening of the skin due to lack of circulation of blood. Next, there's algor mortis, which is the cooling of the body after death. It starts to drop within an hour at a rate of about 1 to 1.5 degrees per hour, and after 12 hours the rate slows. Next is rigor mortis, or the stiffening of the body. Muscle tissues contract because of calcium buildup and no ATP being produced, which is needed for muscle relaxation. It is first noticeable in the smaller muscles like the fingers and toes. It begins about 2 to 6 hours after death, and in about 8 to 12 hours after death, the body is completely rigid, as shown in the figure to the right. After another 8 to 10 hours, it disappears completely due to the cells beginning to break down. Livor mortis, or lividity, is due to blood settling at the lowest point because the heart is no longer pumping. It can be seen as early as 30 minutes after death, and it fixes after about 8 to 10 hours. This means that if the body is moved, the blood will not settle at another lowest point. Next, there's insect activity. This is probably one of the most common types of PMI indicators used. Often, the life cycle of a blowfly is used. Their life cycle is about 2 to 3 weeks. Botanical evidence, or plants, can also be used to determine time of death. Plants on, under, or around the body are assessed by the degree of growth and the type. Bones can also be looked at through their chemical changes to determine time of death. Citrate decreases linearly after about 4 weeks, and assessment of scattering due to scavengers can also help pinpoint time of death. All of these indicators have limitations. For example, algor mortis, or the rate of cooling of the body, can fluctuate if extreme or fluctuating weather is present. The health at the time of the death can also affect these indicators. If your body is in water, it will decompose at about half the rate than if it was not. A lot of these indicators are good for only IDing time of death up to a certain point. If the body has been discovered later in the decomposition process, things like rigor and algor mortis cannot be used. Insect activity also may not start right at time of death, so everything may be shifted. Or if the body is indoors, there may be no insect activity at all. There are 5 main stages of decay. There is no fine line between these stages because decomposition is a continuous process. However, putting decomposition into stages can help narrow down the time of death and classify characteristics. The first stage is the pre-bloat or fresh stage. In this stage, you start to see discoloration of the skin, and you see algor, pallor, and rigor mortis. You also start to see fly eggs being laid. This stage happens about 1 to 3 days after death. The next stage is the bloating phase. This is about 4 to 10 days after death. You start to see swelling, especially in the abdominal area, due to byproduct gases of bacteria. Fluids begin to expel from the body at this time because tissue is being broken down, and it can even explode in extreme circumstances due to pressure. and also the larvae start to hatch. This third stage is the post-bloat or active decay stage. In this stage, maggots and microbes begin to really break down the body, and this happens 10 to 25 days after death. The next stage is the advanced decomposition stage. Maggots have turned into adults and have left the body mostly at this time. Only skin, cartilage, and bones are left, and you start to see a lot more beetle activity. The final stage is skeletalization, which happens 50 days after time of death. Only hair and bones are left, few insects are left, and there's an increase in plant growth due to nutrients that have leaked out of the body into the soil. Next, I will explain the role of microorganisms in decomposition. It is theorized that microorganisms have evolved to recycle vital nutrients from mammals back into the ecosystem. First, before microbes even get involved, cells go through autolysis, which is when enzymes dissolve cells from the inside out. Cells rupture and leak nutrient-rich fluids for bacteria to feed on. This is also what causes skin slippage. Autolysis is rampant in the liver, gastrointestinal tract, brain, and other high enzyme content organs. Putrefaction is because of microorganisms catalyzing tissues into gases, liquids, and simple molecules. These byproducts include lactic acid, propionic acid, methane, ammonia, and so on. Cadaver dogs actually detect these byproducts, and decomposition would not happen without microbes. Decomposition usually begins in the gastrointestinal tract. Bacteria migrate from here to other parts of the body. Staph and chloroform bacteria, followed by anaerobic, are the first to migrate out. As stated earlier, bloating is caused by these byproducts of the bacteria. They create hydrogen sulfide, methane, CO2, ammonia, and hydrogen gas. The skin, outer cavities, and muscle tissues see a shift from aerobic to anaerobic bacteria. This is due to the loss of oxygen in tissues because of no blood circulating. So you almost get a sandwich effect of bacteria breaking down the body from the inside and the outside. Despite their vital role, researchers have only scratched the surface on how microorganisms are really involved in decomposition. In 2013, Hyde and other researchers produced one of the first umbrella reports of what microbes are present through decomposition. 2 cadavers were placed in Texas and were studied through the pre- and post-bloat stages. They sampled the mouth, stomach, and colon. The results showed a shift from aerobic to anaerobic bacteria, specifically staph and enterobacteria to Clostridia and Bacteroides. Metcalf in 2016 showed that decomposition is not a static process. She produced this clockwork diagram to show what types of microbes were found at different points in decay. After finding specific microbes at different stages in mice, she repeated the experiment with human cadavers. She found that as decomposition progresses, there is an increase in nitrogen recycling and amino acid degrading bacteria. In 2017, a study observed microorganisms in the oral cavity of a decomposing cadaver by sampling it daily. They found Formicus and actinobacteria in the fresh stage, Tenoricus in the bloat, and different types of Formicus in the advanced stage. A Clostridium shift was found in a study focused on decomposition microbes in the gut. They found a shift from Clostridium novi to an unknown species. The purpose of my research is to compare microbial communities through stages of PMI of a decaying human cadaver in order to further explore the use of microbes as a forensic method to determine time of death. This could fill in the limitations of other PMI indicators discussed earlier in the presentation. For example, the indoor environments, where there could be no insect activity and after a certain point, things like liver, pallor, and rigor mortis cannot be used. A sub-purpose of my research is to identify certain species that could be easily tracked through decomposition or show up in certain stages. My null hypothesis is that there will be no differences in the microbial communities throughout postmortem intervals of a decaying human cadaver. My alternative hypothesis is that there will be differences in microbial communities throughout postmortem intervals of a decaying human cadaver. Our collections were made at the University of Tennessee's Forensic Anthropology Center, also known as the Body Farm. It was founded by William Bass in 1971. All cadavers are donations as you would donate your body for medical students. We sampled 6 cadavers under natural conditions in various stages of decomposition. We had 1 in pre-bloat, 2 in bloat, 1 in post-bloat, and 2 in advance, with 1 being in a subcategory of mummified. We swabbed in replicates of 2 for each cadaver. We swabbed the left ear canal, right nostril, soil, and air. We chose the ear and nostril because no past research has looked at this area. There is also a distinct field advantage to sample these areas in criminal investigations because it leaves the body undisturbed. Soil and air samples were taken to exclude any microbes in our results that might have come from the surrounding environment. When collecting the samples, swabs were dipped in sterile water first, then swabbed, and after samples were collected, they were placed in collection tubes. Once samples were obtained, they were transferred back to Piedmont to extract microbial DNA using the QIAGEN DNA Easy Power Soil Pro Kit. A modified protocol of the step-intensive procedure seen on the left was used. During this process, a vortex machine was used to aid in the extraction. DNA extraction was used instead of culturing techniques because of the dangers of certain bacteria that would have been cultured and the exclusion of anaerobic bacteria. Whole genomic DNA was sent to UGA's genetics lab for amplification and sequencing. 515F and 186R primers were used along with 2x850 paired end sequencings to amplify the V4 region of the 16S rDNA gene. The V4 region was chosen because it was the most highly variable region of bacterial species. This is important because we wanted to have high confidence in IDing the species present. Illumina sequencing with next-gen techniques was used to sequence the reads. We used the program R to run our analysis. We asked 3 sub-questions to test our overall hypothesis. First, is there a difference between the soil's microbial communities and the body's? Next, is there a difference in the microbial communities between the nostril and ear canal? And finally, is there a difference between the microbial communities by decomposition state? Our data includes 23 samples between swabs from the ear canal, nostril, and controls of all cadavers, and we had 1,001 operational taxonomic reads. We tested the soil versus the body communities in order to rule out any microbes that originate from outside the body. We only wanted to look at microbes from the body to factor out environmental variations. We used a Bray-Curtis dissimilarity, which quantifies compositional dissimilarity between 2 sites based on the counts of each site. Then we did a Tukey's range test, which gave us a p-value of 0.000209. This showed a significant difference between the soil and microbial communities. The graph on the right displays 95% confidence intervals. The fact that the ellipses do not overlap again shows significant differences between the communities. The NMDS plot is another way to organize data. The closer the dots, the more similar the communities. The black dots are soil, the red pre-bloat, and so on. The open dots are nose, and the closed are ear. As you can see, the soil communities are grouped away from all the other body communities. Then you see we have an advanced community over here, some pre-bloat over here, and the bloat are over here. This is a good interview showing the differences between all communities swabbed. The purpose of comparing the ear canal and nostril microbial communities is that we want to see if there was a difference in communities based on the location sampled. We again used Bray-Curtis dissimilarity. As you can see from the image, the ellipses are overlapping, showing that communities have similarities. We also did a Mann-Whitney U test, which gave us a p-value of 0.60378. This tells us that there is no significant difference in the swab locations. The bar graph shows the 95% confidence intervals for the ear and nostril communities. The nostril shows to have more variance than the ear. However, the nostril had less counts per OTU than the ear canal. Next, when comparing our microbial communities by decomposition state, we decided to use Fisher's alpha diversity over Shannon index. We decided to do this because Shannon needs your data to follow a normalized distribution. However, our data follows a logarithmic pattern. We ran a Mann-Whitney U to compare our alpha diversities. Due to our limited sample sizes, we decided to split our samples into 2 groups, pre-bloat and bloat versus post-bloat and advanced. We got a p-value of 0.00338. This means we had a significant difference between the two groups, which supports that microbial communities change through decomposition state. This means that we can reject our primary null hypothesis that there is no difference in the microbial communities. Before digging into the discussion of our results, I wanted to acknowledge our low sample size. This was something out of our control because the FAC only had a limited number of cadavers for us to sample, and we did not know the decay state of the cadavers until we collected our samples. The significant difference between the soil and body communities could be due to the limited exposure of these areas to the soil. If the cadavers had been buried, results may have differed. Results allow us to only look at the body communities with confidence that they did not originate from the environment. The ear-canal and nostril communities could be similar due to them both being external cavities on the face. The nostril could have more variance due to it being a part of the respiratory system. It is therefore exposed to more factors providing different types of nutrients. Difference in decomposition state is most likely due to the change of the body environment after someone dies, as discussed earlier. Bloating is a stage where oxygen begins to get used up, transitioning the environment to an anaerobic one, creating different selective pressures. Also, as bacteria break down the body, different resources become available or not available anymore. As decomposition progresses, bacteria have access to different parts of the body and migrate to different areas. Again, the decomposition process is a vibrant and changing process. I will now go into further detail about the types of bacteria present through each stage of decomposition. Due to time constraints of this presentation, please look through the next 5 slides at your leisure. If you desire further explanation of the particular species found in each stage, my presentation is also on YouTube under Microbial Changes through PMI. Again, the purpose of our research was to explore a mechanism to use as a tool to determine time of death for forensic investigators. We wanted to see if there were shifts in certain types of bacteria that could be used as identifiers for certain intervals. Also, it is not surprising to see some bacteria in more than one interval at different abundance levels, as again, the decomposition process is a fluid process. This graph is based on the class of each stage and location swab. I'm going to go through a few classes that are prevalent. Bacteroid and Aristopelotrachea are seen mostly in the earlier stages. Clostridia really starts to show up in the bloat stages and peaks in the advanced. Gamma proteobacteria is found in the post-bloat stage as well as some in the advanced stage. In the advanced mummified stage, you see Actinobacteria, Plantomycetium bacteria, and alpha proteobacteria mostly just show up here. Our results are similar to Metcalfe's microbial clock, a paper done by Hyde in 2014 and the study done on the oral cavity. Although we did find some anaerobic genuses in earlier stages, there did seem to be more anaerobic bacteria in later stages. The following areas are areas in which this research could be built upon. First, the study could be replicated to increase sample size. Next, significant species identified could be tracked through more points of decomposition. This data could then be used to create a model for these species in order to easily pinpoint time of death. As stated previously, there are many variables to take into consideration when pinpointing time of death. By replicating the study in different environments, the effects of things like water, extreme heat and cold, humidity, and so on can be taken into consideration when looking at microbes through decomposition. Finally, these results could be taken and compared to microbial communities in a living human to serve as a baseline for this data. Thank you, Dr. Austin, for helping me come up with this topic and helping me find ways to make it possible. Thank you, Dr. Nguyen, for all the help you did on my analysis and of the data. We could not have done it without you. Thank you to Professor Willis for assisting me and Dr. Austin at the Body Farm and the valuable resource you are for death investigations. Also, I would like to thank the Forensic Anthropology Center at the University of Tennessee for letting us use their facilities and for the staff members that helped us navigate the property. Also, thank you to the donors at the FAC, as this research would not be possible without their donations. Finally, I would like to thank my friends, classmates, and family for all their support.